Okay, if everyone's done with their do it nows, we'll actually move on to the next step. Today we're going to talk a little bit more about DNA. We're going to further our discussion. We're actually going to do something very hands-on, a little bit Julia Child, so to speak. You'll see I have a whole bunch of ingredients lined up for us to work with today. And today we're actually going to be extracting DNA from peas. So I know that sounds kind of fun. So the first thing we're going to do is add a cup of water, half a cup of peas, and some salt into our blender. And we're going to let it blend for 15 seconds. So everybody want to count? Tell me when we're done. Okay. Now everybody be paying close attention because you'll notice in the back of the room there are a bunch of different items for us to choose from. And you're all going to get a chance to do this with maybe a little bit of modification on your part. So what we've done is we've blended this for 15 seconds and we're going to pour it through a strainer. And you're asking, well, why did we blend the material? Well, what we want to do is we want to break down the cell wall so that we can actually get the DNA out. Okay, so now we've got it in here. Okay, now the next step we have to do, we've got this thin kind of pea soup, anemic pea soup, is we're going to take detergent. I want you all to be thinking about why we're using perhaps the material that we're using. All right, so 2 tablespoons of detergent. And you'll notice also that you have quite a few detergent options back on the lab desks too. Okay, so actually we want to swirl this together. Don't mix it really, really hard, just kind of a light swirl. All right, now you have to let this sit for 5 to 10 minutes. But because we don't have 5 to 10 minutes to wait, through the magic of television, I have some here that I've already prepared. And what we're going to do is we're going to put it into another container. Now that we've strained it, we've gotten all the loose bits out, we're actually going to pour it in. This has been sitting, like I said, for 5 to 10 minutes. And what we want to do is pour it into, you're going to have 3 test tubes back there. So just pour a little bit into each container, each test tube. Don't maybe fill it halfway. Okay, because you're going to have to add some alcohol, so you want to make sure you have enough room. Once again, I want you to be thinking about what exactly we're adding and why to the steps. Now this, you'll see, is a meat tenderizer. Now why in the world would we be using a meat tenderizer? Let's think. Meat tenderizer breaks down particles of meat. So we're using this. You want to just mix it in, but just carefully mix it in, very gently, okay? All right, so you want to do that to all 3 test tubes. And we're doing it to 3 because we want to, you know, sometimes things go wrong, maybe we'll do it too hard in 1. We want to make sure that we've got enough time to make sure for any margin of error, so to speak. Okay, so now that we've done that, the next step that we have to do is we're going to add alcohol to the solution. All right, so we're getting closer. We're getting closer and closer. Now be paying attention because, like I said, we're going to be doing this and you're going to be doing this on your own in the lab. All right, so you want to add about equal parts. You're doing this slowly because you want the layer to form, the alcohol layer to form on top. Okay. Anybody have any questions? Everybody understand? Okay. All right, here we go. Now we just want to let that sit for a minute. So while we're waiting, this is the last step. So we're just going to let that sit for just a few minutes. Let's think for a minute about what DNA is. Now DNA codes for what, Michael? That's right, amino acids. Good answer. And what do amino acids form? Anybody? Jolene? Yes, that's right, proteins. Good answer. Now these are the building blocks of life, so to speak, and DNA is in every living thing. So I know it's kind of hard for some of us to wrap our brains around DNA because we can't actually see it. And that is the point of this experiment. We want you to be actually, to be able to see DNA. And you kind of don't think about DNA being in plants. You know, DNA is always talked about humans. We have fruit flies. But I mean, how many of us have actually thought about the fact that DNA is in the celery that we eat, or, you know, the grapes that we're eating? So we want to think about that. And DNA codes for everything. It codes for what color hair you're going to have, what hair, you know, you may have a disease such as, you know, sickle cell anemia. And mutations are going to lead to things that you might not, you know, that wouldn't be very good for you. So it's important to understand DNA. Now this is kind of, you know, things that you can do in your own home kitchen, so to speak, to come up with DNA extraction. Now in a lab, this would be done, you know, a little bit more high tech. You wouldn't be using laundry detergent and salt. But I just wanted you guys to get an understanding and kind of the basic idea of the fact that DNA is in everything, and it's very important. And the reason we use pea plants today is kind of a shout out to Mendel and his pea plant experiments, which, you know, consider the father of genetics. So now the next thing you're going to do is you're going to take a toothpick, and you're going to notice, I know it's kind of hard to see on film, that you're getting these long stringy things. Now I'm going to come close to the camera so that everybody can see. You're getting these long stringy filaments, and this white goop that forms between the layers is actually the DNA, okay? Anybody have any questions? All right, so the lab you're going to do today, everybody see? The lab you're going to do today, you have a lot of options to choose from in the back. You've got, first and foremost, so I want to make sure that everybody has their safety glasses on and their gloves, because some of this stuff, when you're going to be working with raw chicken liver, I want to make sure that we take full precaution with that, since it's going to be raw meat. We're also going to be working with raw beef, so we've got to be careful, and no eating anything that's here. You have options in the back that range from different fruits and vegetables to initially blend up in your blenders. Then you also have different types of salt. You've got kosher salt, you've got pink Himalayan salt, you've got sea salt, you've got iodized salt. Then you also have to take into consideration the temperature of the water. Now I used ice cold water. There are ice cubes in the bath while it was waiting for you guys to come in. I made sure it was nice and cold. So your job with your data probes is to measure the temperature of the water, of the solution at all points in the experiment. Because we want to see if perhaps the temperature of the water has something to do with how much DNA we get. Now, all three, I've gotten a lot of DNA. Like I said, I know it's kind of hard for you guys to see. You can come up before you go back to your lab station so you can see just how much DNA we've gotten, because this is pretty fun. Science is fun. Okay, so make sure you kind of follow the same steps that I did. So you've got the salt, you've got a bunch of different detergents, you've got name brand detergents, you've got powder detergents, you've got bleach, you know, detergent with bleach. We want to see if any of those might possibly have something to do with a higher or lower DNA yield. And lastly, we've got a couple different types of enzymes. In addition to the meat tenderizer, we also have pineapple juice and contact solution. And for both the pineapple juice and the contact solution, make sure that you're using just a drop or two. You don't need to go overboard on that, because you want the DNA to be formed, the process to be done slowly, because otherwise you're going to run into perhaps a lower yield of DNA. And lastly, I've got a couple different types of alcohol back there. I've got ethanol, isopropyl alcohol, denatured alcohol, you know, 190 and 200 proof. So just make sure, once again, that you follow all the established safety lab rules. And if you have any questions, I'll be moving around the lab, but certainly feel free to ask. The main point of this lab today is I really want you to think about what you can do perhaps to get more DNA, or as much DNA as I did, this method that I did is established. So we just want to kind of veer from the norm and see if perhaps if what I'm doing is the best way to do it. Perhaps, you know, if I made the water hotter, would it have worked better? I'm not sure. I want you guys to think about what each step would actually perhaps be like if you were doing it in a lab. Why do we blend the peas? Why do we add the enzymes? Why do we add the detergent? Why do we add the salt? And just be careful to list all the ingredients that you used and how long you let. Like I had to let the detergent sit for 5 to 10 minutes after I added it. Maybe you want to let yours sit for 15. So, you know, take all that into consideration. Make sure that the recorder in your group writes down the detailed list of what you added and how long it took. I also want you to make sure that person that's recording is also listing the temperature of the solution, like I said, through the entire experiment. I do have hot water, so be careful, once again, not to burn yourself. If anybody wants that, I can help you assist. It's in the tea kettle in the back. And basically, I just want everybody to have fun. Science is fun, science is learning, and don't be afraid to make mistakes. And that's how sometimes some of the best inventions have ever been created, through mistakes. So, you know, just play it safe, but have a great time, and like I said, I'll be walking around the room. If you have any questions, just let me know, and good luck, everybody. Thanks.